Hello, my name is Daniel Montes, and I'm a rising junior MARC fellow at the New Mexico State University. Since high school, I've had a great interest in neuroscience. As a biology major, I want to increase my understanding of how the nervous system repairs after injury or disease. My current plan is to enter a graduate program that focuses on neuroscience, so I joined MARC to gain lab experience. I joined the laboratory of Dr. Graciela Unguez in the biology department. Dr. Unguez has been interested in how different electrical patterns affect the plasticity, differentiation, and regeneration of neuronal circuits in vertebrates and has been using different electric fish species as a research model system. Most of the electric fish that we work with are indigenous to regions around South America and Africa. These electric fish generate an electric field around them to communicate with other fish and to navigate in their environment. The field emitted is not used temporarily by the electric fish, but constantly. This constant electric field is generated by a special organ in the electric fish called the electric organ. The electric organ is composed of many cells called electrocytes. The electrocytes have a shape similar to a cigar. The electrocytes are derived from the skeletal muscle fibers by the fusion of multiple muscle fibers. They are bigger and capable of sustaining constant firing of 50 to 200 Hz of electricity, whereas the muscle fibers are much smaller and can only sustain less than 8 Hz of electricity intermittently. A heavy supply of electricity can destroy a cell. When an electrocyte does not have enough, it dedifferentiates back into a skeletal muscle cell. Here's a short video of what the electric field emitted from the fish sounds like. It is fascinating how these electrocytes can be electrically active at such high frequencies 24/7 throughout the life of the animal. Another very unique feature of these electric fish is their ability to regenerate lost body parts. Most vertebrate animals have very limited capabilities in restoring lost tissues. These electric fish are amazing. When their tail is amputated, they will regenerate all the tissue in the tail portion that was lost, and this can be repeated many times. The regrown tail is not just certain cells or tissues being regrown, but all of what was in the original tail. We would see the skin, muscle fibers, electrocytes, bone, spinal cord, and blood vessel all be regrown. Previous studies in the Unguez lab show that the main process of regeneration in one electric fish species is by the activation of stem cells associated with skeletal muscle cells and electrocytes, similar to how our own muscle cells are repaired after injury. My research project will focus specifically in the variation of the stem cell count after subsequent injury. We are interested in characterizing the satellite cells to get a better idea of how these fish regenerate and how these cells differentiate. To do this, we perform immunofluorescence, a technique used to label specific biomarkers, like proteins, that are only present in specific types of cells. In my research, immunofluorescence is used to label transcription factors only found in the nucleus of stem cells we are looking at. It is known from previous studies that the stem cells are found along the periphery of the electrocyte and muscle fibers, so our markers should only illuminate around the edges of the electrocyte and muscle fiber cells. As stated previously, satellite cells are necessary for the tail regeneration of electric fish. Whether subsequent injury affects the amount of stem cells in the regenerated tails of the electric fish is unknown. That's what my research is trying to conclude. To conduct my research, I'll be working with the Synophagus macrarus, analyzing the total number of stem cells present in tissues after multiple injuries. I am interested in knowing if the total number of stem cells associated with the electrocytes and muscle fibers decrease after subsequent injury. Is there any use or benefit that my research can offer? For the benefit of humanity, this research can provide further insight into injury recovery and tissue regeneration. By having greater insight of the stem cells' roles in the fish regeneration, we can use the implications to understand how stem cells play a role in aging and injury recovery in other species, including mammals like you and me.